Hello everyone, in this video I'll be talking about diffusion and osmosis, but in correct terms I'll be talking about the transport in cells, whether inside or outside the cell. But to get to there we have to first get the idea of diffusion, and later we'll move on to the more complicated idea of osmosis. Now you all know that if we spray a perfume in one corner of the room, it will start spreading around until, with time, until everyone can smell it, even those at the other corner. Even without spraying it or giving it that push, if you just open the bottle and leave it there, the perfume will spread around by itself around the room. Are we giving it any force to spread around? No, because that's a natural process that occurs, which is diffusion. So if you see here, if I put a crystal dye inside of a fish tank, and this is more like a magnified, super magnified picture of that rectangle, which shows that the concentration of the crystal dyes here is in this area. If I take this area and I ask you how much or how does the crystal dye concentration compares in this area and any area here, of course this is a very high concentration of crystal dye, and this is very low to none concentration of crystal dye. And with time the crystal dye of course will start to spread around from an area of high concentration to an area of low concentration until it's equally distributed, which means if I take a rectangle here or this one, I will find the same amount of crystal dye spread out in the same way. Does that mean they're not moving? No, they're still moving, but we say they are equal distribution or they are in equilibrium. Now when I say the word concentration, for anyone who does not know concentration, concentration is calculated by this formula: C equals N over V. N being the number of particles, number of moles or number of particles, and V being the volume. So it's not just how much particles you have in a solution, or how much solute you have in a solution. Here we'll be talking about solution. So let's say, this is the example I usually give. Let's say you have a classroom with 100 students, and you have a whole city with 1,000,000 students. Which one do you think has more concentration? Of course it's the classroom with 100 students, because it's not about how many students or how many particles you have. It's about the distance between each and the other particle. So here they'll be very spaced apart in the city, and here they'll be all together in one classroom. So when we talk about concentration, we have to take into consideration the number of moles, or the number of particles, and the volume. So if I have a concentration of, let's say I have water, and I've put 2 spoons of salt here, and now I have a solution containing salt that's dissolved in it. If I ask you how can I increase the concentration of this solution, there are 2 ways. I either add more salt or decrease the volume by, let's say, evaporating the water. If I decrease the volume, which shows in this formula, the volume is inversely proportional. So if I decrease the volume, the concentration will increase, because the particles of salt will have less space or less volume to spread around, so they'll be more squeezed with each other, which causes more or higher concentration. So this is just to sum up the idea of concentration for anyone who may be confused with this. So this is the definition of diffusion. It's the movement of molecules or of particles from an area of high concentration to an area of low concentration. We say down the concentration gradient. The gradient means the difference. So if I have high concentration in here and low concentration here, I do have a gradient, and the particles will move down, so from high to low. Not until all of them are here, but until we reach what we say equilibrium. That's diffusion, and it's only particles, or in this case we'll be dealing with solute, so not the solvent, which is the water. When it's water moving, that's osmosis, and we'll see it now. Now when it comes to the cell transport, there are two types of cell transport, either passive transport or active transport. The passive transport is the one that does not require energy, but the active transport will require energy in the form of course, ATP. The passive transport, we can find diffusion, facilitated diffusion, and osmosis. Diffusion, um, all of these are from high to low, from down the concentration gradient. If we want to say to look at diffusion or simple diffusion in the cells, as I showed it before, it's the distribution of molecules, and as you can see here, if I have high concentration, if this is a cell, I have high concentration of a substance outside the cell and low inside. The substances will start to move in until they are equally concentrated outside and inside. It's simple diffusion when the substance can easily pass through the cell membrane without the need of any help. So they can squeeze themselves between those phospholipids until they are equally spaced. Examples of these molecules are oxygen and CO2. Oxygen and CO2 can just pass through the cell membrane, whether in or out the cell. So they don't actually need help. Facilitated diffusion is still from high to low, but these molecules are either big, that they cannot pass through the cell membrane, or they are polar, or they are hydrophilic. And as you know, this area of the cell membrane is hydrophobic, so these cannot pass through. This is why they might need a channel protein, which will allow them to go in or out, or such as this example, it's a receptor protein that will help move them around. So facilitated diffusion is very similar to diffusion. It's using transport protein. Active transport is when you're going against the gradient, so you're using energy. As you see here, we have the high concentration here, the low concentration here. So the normal case is these moving to the other side. But in the active transport, the cell would want to keep something very important in, or something very toxic out. So it will use pumps or transport proteins to actually pump these molecules inside using the energy from ATP. So we're going against the gradient. We're causing even more high concentration and more low concentration here. So what's osmosis? Now we get to the osmosis part, which is the third example of passive transport. So if I want to move here, so concerning osmosis, if I want to explain osmosis, so let's say we have this beaker, and it's filled with water, and we have something separating these two sides, which is a semi-permeable membrane. Now a semi-permeable membrane will allow few things to pass through, but not everything to pass through. And I have here high concentration of a substance, let's say salt, sugar, and here very low concentration of a substance. Now the first thing you look at is that, okay, we have high concentration here of the solute and low concentration here, so these would probably diffuse from here to there. But since this is semi-permeable, these molecules cannot pass through, so they would bump back, and they can't pass through. So we have a difference in concentration here. We have a problem, and we cannot change the number by bringing the particles from here to there. What's another thing to change to change the concentration is the volume. So what will happen, since it's semi-permeable, it will not allow the solute, but it might allow the water to pass. So the water will go opposite to the solute. It will go from low, opposite to the solute gradient, from low solute to high solute, and we'll end up with something like this. What we did is that we decreased the volume here so that the concentration increases, and we increase the volume here so that the concentration decreases until both concentration on both sides are equal. Now this is osmosis. So if I put it in percentage, let's say I have here 20% solute and here 10% solute, and the same, the membrane is semi-permeable. So instead of diffusion happening from high to low solute, osmosis will happen and water will move from low to high. Now if I have 20% solute, that means 80% water, and 10% that means 90% water, which means that osmosis happens from high water to low water. Now, so that you don't get confused, you can always look at the solute and you go from low to high. But just in case the question mentioned how much water you have, so let's say it's mentioned that you have 95% water and 75% water here, then you can calculate the solute, which is 5% solute and 25% solute, assuming this is semi-permeable. So the first thing you think of, okay, semi-permeable, then it's osmosis. Then you go from low solute to high solute, and that's it. You need to know first what's the process happening. If it's permeable, it's diffusion. If it's semi-permeable, it will be osmosis. Then you look at the solute and you decide if it was permeable, if it was diffusion, then it goes from high to low solute. If it was osmosis, it goes, the water will move from low to high. You need to mention that it's the water moving, not the solute, or else it's wrong. If you don't have the solute concentration, you have the water concentration, you can just calculate the solute concentration. Now we get to three main terms. So let's say I have solution A, which has 20% solute, and solution B, which has 10% solute. Solution A has more concentration than solution B. So I say that solution A is hypertonic to B. Okay, so A is hypertonic to B, but B is hypotonic to A. So the word hypertonic means having more solute, hypotonic less solute, and the word isotonic will mean having the same amount of solute. So if I put a cell, if I told you that I put a cell, we come to the cells now. So I put a cell in a solution that has 20% salt and inside a 10% salt. Is this cell put in a hypertonic or a hypotonic or an isotonic solution? So we're comparing the solution to the cell. We say the solution is hypertonic to the cell. If the solution is hypertonic to the cell, then, and we assume always that the cell membrane is semi-permeable, so what will happen in this case? The water will move from low solute to high solute, which will cause the cell to shrink or shrivel. Now if I put a cell in a hypotonic solution, so there is much more solute inside the cell than outside, having the cell semi-permeable, the water will move from outside the cell to the inside until the cell will expand or even blow up. If it's an animal cell, if it's a plant cell, it will just expand because the cell wall will protect it from blowing up. So first thing you check, is it diffusion or osmosis based on the membrane, is it semi-permeable or permeable. Second thing you check is the percentage of solute or the gradient difference in solute. If it's diffusion, you go from high to low, the particles moves from high to low. If it's osmosis, the water moves from low to high concentration of solute. I hope this is helpful and will help you in your study. Thank you very much.